esophageal cancer continues to be very challenging. I would say, a lot of patients will present with metastatic disease, and that could be either after they've been treated previously or just kind of new onset. And at that time, usually they get first-line chemotherapy. And once you do that, any other treatment you give them is just less effective. Unfortunately, a lot of those chemotherapy drugs have kind of cumulative side effects. So if the first-line regimen gives you neuropathy, your second-line chemo regimen also gives you neuropathy. So not everybody actually qualifies for the FDA-approved second-line treatment. It's not an easy regimen to tolerate and has a lot of side effects. So it is an option that we discuss with patients. Definitely, whenever we present a clinical trial, we start by discussing the standard of care. But this trial will definitely be a second-line option that we will discuss as well. Not everybody will qualify for this trial because even though it's mainly an esophageal adenocarcinoma clinical trial, it's actually only a specific subgroup of patients with esophageal adenocarcinoma that could go on that trial. And those are patients that on genomic sequencing, when we analyze and catalog the genetic targets of their tumor, we find that they have specific abnormalities in a bunch of genes. And if they do, or if their tumor shows a lot of genome instability, then they could qualify for this kind of a DNA repair inhibitor. We will analyze the genetics of the tumor, and patients have to have an abnormality in a pathway called homologous recombination. That's a DNA repair pathway that we all have that plays a major role in us repairing damage from multiple things, because as we live and breathe, as we age, we have, whether we smoke or not, we are getting exposed to constant DNA damage. And in people with esophageal cancer, they don't inherit a genetic change that increases the risk of esophageal cancer, but their tumors develop genetic changes in either one or multiple proteins in that same pathway. And that actually makes them highly more sensitive, potentially, to this drug called niraparib, which is known as a PARP inhibitor. So in this clinical trial, we're really first, as a primary objective, we're trying to see if patients with esophageal cancer that harbor an abnormality in those homologous recombination genes, do they respond to niraparib or not? There is in vitro data, tumor cell line data that suggests they should, but you never know if that translates into the patient benefit. And then we're also assessing the usual: do they live longer? Do they live longer without cancer progressing? Do they tolerate the drug? How does their bone marrow do with those patients? And in addition to that, we're trying to know if you do respond and then you stop responding, why did you stop responding? So we're doing some of those translational correlative analyses to actually define what are the mechanisms of potential resistance to those drugs. We try to analyze the genetic changes and how they associate with treatment in that same pathway. We actually also assess something a bit different, which is called loss of heterozygosity. That is kind of a measure of overall genetic instability. And some of those esophageal cancers are very genomically unstable. So we're actually going to do kind of what they would call a genome-wide analysis of loss of heterozygosity in tumor tissue and try to see if that by itself could be a marker for response to niraparib. We're also going to look at some of what we call reversion mutations. Unfortunately, what happens sometimes is you have a genetic change, we give you a drug. Yes, we inhibit the impact of that genetic alteration or loss of that gene, but then you develop another mutation that makes you resistant to the drug. So they're called reversion mutations, and these have been identified in certain cancers, and so we are actually trying to study whether the same phenomena happens in esophageal cancer.
The purpose of this study is to see if participants with metastatic esophageal cancer respond for a longer period with niraparib versus standard of care treatment. This is a phase 2 single-arm, 2-stage study. In stage 1, 18 patients will be enrolled. If 3 or more patients respond to treatment, the study will continue to stage 2. If the trial goes on to stage 2, 25 additional patients will be enrolled. Study participants will be treated with niraparib until their disease worsens or they experience severe side effects. Niraparib is a type of drug called a PARP inhibitor that kills tumor cells with certain genetic mutations. To be eligible for this trial, you must have a homologous recombination or HR mutation in your tumor tissue, a high rate of loss of heterozygosity or LOH in your tumor tissue, or a germline mutation in your blood. These mutations can affect the way your cancer responds to certain treatments and are common in patients with your type of cancer. You may be eligible for this study if you have cancer that started in your esophagus, GE junction, or stomach that progressed and spread to other parts of your body after receiving a platinum-containing chemotherapy regimen. There are other criteria that must be met to be eligible for this study. Talk to your doctor to learn more. To learn more about this clinical trial, go to HoosierCancer.org and choose Current Trials from the Clinical Trials Navigation tab.